Our next speaker is Dr. Dawn Branch, a professor in medicine and laboratory medicine and pathobiology at the University of Toronto, and a senior scientist in the Centre for Innovation with the Canadian Blood Services. Dr. Branch has published more than 210 original research papers and has received numerous awards of distinction for his work in transfusion and transplantation medicine. Dr. Branch will be speaking to us about intravenous immunoglobulin mimetics and replacements. Welcome.
Good morning, and thank you for that deserving introduction. I'm really happy to be here, and I hope you enjoy this presentation. I spent a lot of time on it, believe it or not. So my talk is about intravenous immunoglobulin, so I thought I need a little bit of introduction. So I'm sure you all know that IgG is prepared from tens of thousands of plasma donors, and it's all made from human plasma. Sometimes up to 60,000 donors are in one lot. So it's used to treat, as you know, immune deficiencies. That's how it was first developed. So that's a low-dose administration, sometimes up to 800 mg per kilo per month, and as low as 400. But it's also used to treat a plethora of autoimmune diseases or inflammatory diseases, and there it's used at a very high dose, up to 4 grams per kilo. And initially it was used to treat ITP. It was basically discovered to work in ITP serendipitously, really, by Paul Imbach in Switzerland in 1981. But he found that a high dose was able to ameliorate the platelet count, so the platelet count went up, and it's been used ever since for treating ITP. It has a good safety track record, so since 1981 it's been used in millions and millions of patients, and it's been used at high dose. So, like I say, up to 4 grams, and you're probably wondering, who gets 4 grams per kilo? Well, that's a disease called Kawasaki disease, and they give 4 grams per kilo in that particular disease, at least in Toronto. So it does have some side effects. They're relatively minor, except we've seen patients with hemolysis after IVIG, because IVIG contains all IgG, so it has IgG anti-A, IgG anti-B, and very potent anti-A and anti-B. So patients that are A or B can have issues, and in general, about 35% of patients that are A or B will have some level of hemolysis, and that was published by Pendergrast and myself quite a while ago in Transfusion. They also sometimes get thrombosis, but the hemolysis is probably one of the biggest problems. And we found that about 9% of all the patients out of that 35% actually got severe hemolysis. And in that particular group, we found that people that were AB, basically there was a 90% chance of them hemolyzing severely if they received IVIG with anti-A and anti-B. And our work actually led to the Canadian Blood Services using isoagglutinin reduced IVIG as their primary IVIG. So IVIG use in Canada, there's a little bit of history here as well, but about 35 or 40 years ago, the high-dose IVIG was used mostly for ITP patients. And because of that, companies, they always want to make money, so they decided that they were going to develop some other potential treatments. So anti-D was actually early on as a treatment for ITP. Rituximab, TPO receptor agonists, tyrosine kinase inhibitors, daratumumab is a more recent one, and it goes on and on. But a lot of new therapies were developed specifically to treat ITP. But over the past 40 years or so, IVIG not only is used for treatment of ITP, but for all kinds of neuropathies, including myasthenia gravis, CIDP, and others, mostly severe neuropathies, even stiff person syndrome. I don't know if Celine Dion got IVIG, but it is used to treat stiff person syndrome. And it's also used in a lot of autoimmune disease, including warm autoimmune hemolytic anemia, and as I pointed out earlier, 4 grams per kilo for Kawasaki disease. So CBS supplies the IVIG free of charge. Here you go. except in Quebec, which have their own blood system, as we all know. One dose of IVIG, 2 gram per kilo dose, costs roughly $10,000 per dose, and it consumes about 30% of the total CBS operating budget. So you can imagine that CBS would like to get rid of IVIG, so they don't have to pay so much out of their budget for it. So this is kind of an old slide, but just to give you an idea from the University Health Network, the percentage of IVIG that's used for different diseases. So you can see that CIDP and MG are the most common use. Antibody-mediated rejection, multifocal motor neuropathy, but you can see it's actually not used that much for ITP compared to the neuropathies. So that's the big use really now of IVIG. So you might wonder, well, if you're going to replace it with something, you have to know how it works. So since 1981, we still don't know what the mechanism is. So it's actually worse than HIV vaccine, which has been 50 years. We still don't know what HIV vaccine, or we don't have one. Actually, it's about the same time, because I think 81 was when the first immunodeficiency syndrome was described by Gottlieb. So it's the same time frame. Still don't know the mechanism. So how many of you know basically the structure of an IVIG or an IgG molecule? Yeah, how many? Come on, everybody, right? No? Ooh. Okay. Well, think of it as a Y, okay? Why do you want to think of it that way? Because that's what it looks like in a two-dimensional diagram. So you have these two ends up here. They're called FAB, and then you have a bottom part. It's called FC, okay? Does anybody know who first described that and got a Nobel Prize for it? Well, don't sit on a committee with me, because that's one of the questions I always ask a student, is who won the Nobel Prize for the work you're doing? Anyway, it was back in '72, Edelman and Porter. What they did is they were playing around with enzymes in IVIG, or not IVIG, IgG, and they found that certain enzymes would cleave the IgG into different fragments, okay? So that's where you get the term FAB, fragment antibody binding, and the FC, fragment crystallizable. So I just wanted to give you that because we're going to talk about FC. And the only thing we really know about IgG or IVIG and how it works is that it requires the FC gamma. So how do we think it works? Some people think it works by blocking activating FC gamma receptors, which there are 4. These receptors actually cause phagocytosis by activating tyrosine kinase. So maybe they bind and activate 1, 2A, 2C, or 3A, and that causes phagocytosis of the antibody-coated platelet or the antibody-coated red cell. Some people think that it binds and activates the only inhibitory FC receptor called FcR2b. That actually activates a phosphatase that dephosphorylates the tyrosine kinase, so there's no phagocytosis. And then, this is my favorite theory, may bind to the FC receptors and produce an immunomodulatory cytokine effect, like a cytokine storm. And that cytokine storm with IVIG is actually immunomodulatory, down-regulating. So TGF-beta, IL-10, and BAFF inhibitor. So I guess the current thinking really is that it blocks the FC gamma RN, which is the neonatal FC receptor that traffics IgG and albumin so that it gets recycled. But if you block it, then things get degraded, so you can get degraded IgG and albumin. So that's kind of how it works. So what happened here? So they developed strategies, mostly small companies at first. So there are strategies that are being developed or thought about or have been developed that are trying to mimic the immunomodulatory effects of IVIG. So the way they're doing that is with FC gamma multimers. We'll see some of those. They want to block FC gamma RN, Or my favorite is to develop an engineered recombinant IgG antibody library that's multidiverse with mixtures of recombinant IgGs, all derived from human B-cell donors, so that you actually get a recombinant IVIG. So here's a slide of all the different FC multimers that have been investigated, and you can see all these different companies working on their different FC multimers. So here's a hexamer, so you see 6 FCs, another one with 6. This is a stratomere. This is a trimer. So there's all different versions. Now, what's happened here is all of these are now terminated. So we don't have to worry about them anymore. They're not being further developed. But I want to show you one that we've worked on, which is called CSL777. So if you look here, you can see that we were looking at IVIG and this recombinant CSL777. And what we've done here is this is an animal model of ITP. So this is the platelet count on the y-axis. These are the days that we followed these mice. And what happens is we're giving an antiplatelet antibody every day, and we keep increasing the concentration every day so we can keep this nadir down. And then we treat the mice at day 2. So you can see that the recombinant CSL77 increases the platelet count, as does IVIG. So they're very similar. We wanted to understand a little bit more about the mechanism of CSL777. So what this is here on the left, A and C, this is monocytes that are incubated in the cold with a fluorescent labeled, two fluorescent labeled molecules. One here is IVIG, labeled with a fluorescence label, and this is the CSL777 with a fluorescent label. And then the second label is just to the membrane using an antibody called F480. So the interesting thing here is you can see very little kind of binding here actually at 4 degrees, and you see lots of binding here. So the first thing is that CSL77 binds much better to the monocyte. And this is after moving these cells to 37 degrees and trying to look for phagocytosis. You can see that we see some here. This would be the green. This is the labeled IVIG, so it's going inside the cell. And here you see lots of it going inside the cell, and it looks like it's falling apart. We actually showed by Western blot that what was happening is the CSL77 was binding to its receptor. It was then being internalized and completely degraded. So that's why we think it works better. And this is looking at an arthritis mouse model, and you can see this is just the control, which is human serum albumin. You can see the highest we could go for the clinical score is 12. So we let these animals get up to almost 12, and then we start treating them. And you can see that there's not much difference in the human control, serum albumin control, and you can see that CSL777 actually ameliorates almost completely the arthritis. So that's why these were promising, because all of those other ones I showed you, they all do the same thing. They've all been shown in animal models to work similarly as to CSL77. Okay, so then the reason the companies gave up pretty much that is partly because they were sold, the small companies sold to large, big pharma. Big pharma looked at their portfolio and said, we're not going to make too much money doing this, we'll go that way. So they basically gave them up. I mean, they weren't doing badly in phase trials, there were some adverse events, but they could have kept going. But what they decided to do is let's focus on blockers of FcRn. So here's the companies now that are working on FcRn, some of the same ones that were working on the multimers. So what's the current status of that? Well, some of them dropped out, and most of them are still in the business. So one is approved from Argence. It's approved in Japan for MG and ITP use, and it's approved in the States for MG and CIDP. So it's apparently working fairly well. Then they have phase 3 trials for other autoimmune diseases, and then they have a phase 2 that's just started. UCB has a monoclonal antibody to the FcRn that is approved for MG. Most of these companies now are focusing on myasthenia gravis, and so you see a lot of MG on these clinical trials. The only one that's really looking at ITP, the ITP trial wasn't approved in the United States, but you can see that the J molecule is actually being looked at for warm autoimmune hemolytic anemia, hemolytic disease of the fetus and newborn, and fetal and neonatal alloantibody immune thrombocytopenia, FNAIT. So I kind of liked this when I first saw this work. This is a proposal on how to make recombinant monoclonal IgG with high diversity. And it was started by a company in South San Francisco called GigaGen. And if you look on the right side, they basically isolate IgG from normal human B cells, and then they make libraries from those, and they use thousands of donors, and then they can do a massive production of polyclonal engineered IVIG. So this looked promising. It's published in 2021. This company was bought up by Grifols, so we'll see if they move it forward. But the other thing that a lot of people like about this company's process is they can make targeted monoclonal antibodies. So you can immunize somebody to whatever you want, isolate those B cells, show they have the antibody, and then engineer it the way they want. And then you have a product that's a monoclonal antibody to whatever the target was. This is actually how the first monoclonal to COVID was produced. I don't know if GigaGen did it. So if you want to move on here and specifically treat, say, immune cytopenias, which would be involving phagocytosis, then your strategies would be, well, how do we inhibit phagocytosis? Do we block the Fc gamma receptors, or do we inhibit signal transduction with small molecules? So here are a number of companies that are working on targeted monoclonals to Fc receptors, 1 to 2B, 1 to 2A, and 1 to 3, Fc receptor 3, and there's patents applied for that aren't on this list. So there's a lot of people interested in blocking Fc receptors with monoclonal antibodies, including CBS. So this is a figure from a paper from Alan Lazarus's group, where they made a single chain antibody here with an albumin instead of an Fc region. This is the FAB part. And they also made a full single chain with the Fc and the FAB part, and then they just used the whole antibody. And so what they found, and it's published in Gonzalez et al., is that the single chains both worked pretty well to ameliorate ITP, and this one caused some adverse events. So this one was mutated a little bit in the Fc, but it's only single chain. So they're still working on these antibodies. This is antibodies to FcR3, but they also are working on antibodies to gamma R1. So small molecules, what do we have here? Well, I'm going to start at the bottom. So Bruton's tyrosine kinase inhibitor. Bruton's tyrosine kinase is involved in B-cell antibody production. So if you inhibit it, you'll inhibit the antibody, and of course we're talking about autoimmune antibodies. Then there's one called SYK inhibitor, which is spleen tyrosine kinase. That one is approved, and it's used to inhibit phagocytosis, and it's used mostly in ITP, and it blocks the phosphorylation through the activating Fc receptors, so you don't get phagocytosis. And then there's one that we're working on that's called KB208, and interestingly enough, it activates a phosphatase, a serine phosphatase called PP2A, and it's in preclinical development. So I'll just show you a little bit of that, and I'll be done. So inhibition of in vitro phagocytosis using KB208. So this is a mouse model of ITP again. So what we've got is we've got phagocytosis, but we're looking at inhibition, so we're developing an EC50 kind of a curve, or an IC50. And you can see here KB208, it's the square. It's got a nice sigmoidal curve, and it has an IC50 of about 4 micromolar. So it worked very well. This is just showing, again, using it in an ITP mouse model. You can see that if you look over here, the blue bar, you don't have to look at the left side, but the blue bar is IVIG, and this is they're different mouse strains. So this is BALB-C, this is C57 Black 6, and you can see that IVIG is working, I guess you'd say the best, but it's not really significantly different than the KB208 at 2.5 mg per kilo here, 2.5 mg per kilo here, versus 1 gram per kilo here of IVIG and 2.5 grams per kilo of IVIG in the C57. So it works as well as IVIG, but at a much lower dose per kilo. So in summary, many companies and scientists are investigating different ways to mimic IVIG, and Canadian Blood Services does support that kind of research. Problems have been encountered, though, in phase trials with FC multimers. One trial, this is an old slide, one trial is still going, but that's not true anymore. Glitnet was sold to Pfizer, and Pfizer terminated the work. So this is how that multimers have been going, is none of them are now in clinical trials or actually in development. And then, of course, the efficacy of FcRn blockers. I'd say it's still uncertain, but the companies that are trying to make these are trying to make them mutated so they don't bind albumin, because you don't want albumin to be left in the circulation because it carries stuff like low-density lipoprotein. You don't want that to build up. So you want it to be out of the circulation. So my last summary slide is just things I think about that I don't have the money to make. Antibodies to FC receptors, I think if they're going to be effective, they need to be at least bispecific. You can't have an antibody to FcR3 when you know FcR1 also phagocytoses the same antibody-coded cell. So the ideal way would be to make it bispecific, so that you'd have an antibody FAB to 3A here and an antibody to R1 here. But the best way to do it would be a trispecific antibody, where these would be monoclonal, and you'd have one of the FABs to 3A, one to R1, and one to 2A. And the reason 2A, because we've published that certain antibodies actually interact with FcR2A. All of your anti-A, anti-B go through 2A. We have anti-CD47 that causes hemolysis. That's off the market now. But it also went through 2A. That's how it induced phagocytosis. So, and now we're doing some studies with a number of alloantibodies to red cells, and we're finding that probably FcR2A is a very important receptor for phagocytosis in transfusion medicine. So this is what I'd like to see. I'd really like to see Grifols follow up with Gigagen and try to make this diverse IgG. You know, if you want to treat immune deficiency, you could probably have a list of the different pathogens you really want to protect the kid from, or the immune deficient person from, and just make those targeted and then mix them. And now you've got a prep that you could use for immune deficiency that covers all of the major pathogens without having to get hundreds of thousands of donors to make this recombinant. So having done that, thanks. Probably over, but close enough.
Thank you very much. Are there questions in the audience?
Thank you. It was terrific. Do you have any, maybe just a brief overview of the glycosylation of the FC part? How much role that would play or plays in any of that? The N-glycan.
You're asking about the FC?
Yeah.
And what role it plays?
Yeah. I mean, is there any role in, developing molecules with or without glycan? Does it make any difference?
There are companies making FC fragments for treatment. Most people in vitro or in vivo animal models find that FC fragment by itself doesn't really work very well. And that's probably because you need some glycosylation. As far as I know, There are amino acid sequences that have been shown to allow for tighter binding to FC receptors, and that would be something that should be looked at if you're going to engineer an FC, is to make it bind a little tighter. But that's about all I know. I mean, it's important. I mean, all of these autoimmune diseases and inflammatory diseases that I showed you that can be treated, at least by, and lots of off-label use. I don't think they're filling out the same paperwork that was mentioned earlier. They're just using IVIG for almost everything when things fail, right? But it's been shown that if you take off the FC off of IVIG, it fails to work. So FC is really critical, but as far as what sequences might be ideal, that really hasn't been looked at.
Thank you.
Don, what's the status of CSL 777?
It's terminated.
Oh, it just stopped. It seemed to look promising.
Like I said, almost all of those multimers look great in mouse model.
Oh, I see. I missed that. I thought that it was a small molecule.
Oh no, the small molecule is KB208.
I see.
That's still in the works.
Thank you.
But that's a CBS molecule.
Right.
Thanks.
Okay, thank you very much.